Hello and welcome to the overview of the DNA Methylation Analysis module. To begin, you should have watched the Introduction to DNA Methylation video. The introduction section of this module first dives into a little more detail on that, as well as provides flashcards, as you can see here, with some DNA methylation concepts for your review. Here we can see the analysis architecture for differential methylation analysis that is followed in this module. Next, we see a more detailed pipeline that shows the NextFlow NF-Core MethylSeq pipeline that converts the raw reads to the methylation coverage file. First, we begin this module by installing the required packages for this module and loading them. Next, we read the methylation call files and begin to design the experiment. The next step will allow us to see some basic statistics about our data, and then filter based on coverage, as you can see here. This is important specifically when working with samples with overamplification or PCR bias that can affect the downstream analysis. Next, basic normalization is performed to allow us to properly analyze the coverage values of the samples between each other. We then merge the samples into a single table for easier analysis. We then filter the CpGs that are not providing any information about the differences in samples, allowing us to focus on the CpGs that are important. We then move into the correlation between samples and obtaining the Pearson correlation coefficients to assess how samples correlate with one another. Further, we perform a clustering analysis of the distance measures from each sample's percentage methylation. Then, a principal component analysis is performed to assess clustering and variation of the samples in multiple dimensions. Then, as a part of differential methylation, we perform a Fisher's exact test to compare the hyper and hypomethylated bases. We then can perform a beta binomial distribution-based test along with a logistic regression-based test to compare variance in the methylation levels. To further explore the results, we can then see an overview of the percentage of hyper and hypomethylated CpGs per chromosome. Finally, we go through how to perform CpG annotation, which helps in the interpretation of the data at the biological level. Additionally, we have some optional steps and some useful links. Thank you for listening to this overview of the DNA methylation analysis module.